Click for IB Biology. Our topic is genetics, inheritance genetics, and we're now moving on to look at meiosis, particularly from a higher level biology perspective. And in this particular video, we're just tracing the events as seen from the chromosome level. There's a blank version of this presentation, a PDF, down in the description. You can download that, add your own description and notes as we go along. And then at the end, there's a 10-question quiz with answers that you can use to track your own understandings. So I'll just make a slight adjustment to the screen. So there we go. That's just about right. There we go. Okay, so the first thing to pick up on with meiosis, in the images that I'm going to show you, I'm going to be focusing on two homologous pairs, as this represents the kind of limit of the higher level course, and we're looking at it from the perspective of the observable events that can be seen as a cell goes through the divisions of meiosis. And of course there are two. There's meiosis I followed by division meiosis II. So in the interphase, the nucleus is intact, and in fact we wouldn't be able to see the chromosomes in the interphase, but of course we know that during the S phase, each chromosome will be replicated to form a pair of sister chromatids, as illustrated here, and each pair of chromatids is held together by a small structure known as a centromere. So in the image that you can see, we had started with a diploid number of 4, and now this is represented following DNA replication as 4 pairs of sister chromatids, and they become visible during the prophase, and the homologous pairs begin a process of alignment, which is called synapsis, and I'll describe more about synapsis in later videos. So each homologous pair begins to align with each other in a process called synapsis. The nuclear envelope is breaking down, and then this gives way to the formation of the spindle structure, and at the metaphase, the spindle has orientated the homologous pairs to the equator of the cell, which I'll just put in with a dotted line here like that. And so with the alignment, it's now possible to begin the contraction of the spindle structure so that we can separate the homologous pairs at the anaphase. So the separation, as we can see here, with separation of each of the homologous pairs, and this particular action is known as reduction division. And the significance of this first phase of meiosis, once we get to the telophase where there are 2 nuclei reforming, is that we have separated the homologous pairs, and in doing so we have separated the alleles of every gene. This is very significant for inheritance genetics calculations. Having completed meiosis I, each of these cell structures here will progress into meiosis II. Now, it is the case that in some cells, cytokinesis does not actually complete, but these details will be shown as we go through various forms of meiosis, particularly with respect to oogenesis and the formation of egg cells. But those details will come later. We have 2 cells at the beginning of prophase II. There's no particular delay or interphase between prophase meiosis I and meiosis II, so we're straight into Prophase II and Meiosis II, in which we're going to see the separation of the chromatid pairs. And so we expect to see those two purple chromatids separated and the two blue ones. Okay, so as we go into Prophase II, the chromosomes are condensed. In Metaphase, the spindle has formed at the end of Prophase II, and Metaphase II, that spindle structure has now aligned the pairs of chromatids across the equators of the cells. As we move into the Anaphase, we can see here that what we're getting is the separation of the chromatids, so that there's a single chromatid, now called a chromosome, for each of the pairs at each of the poles. And so we can see that we've got to here. So by the end of this event, what has happened is that we have separated the homologous pairs, we have separated the alleles of each gene in effect, and the chromosome number has been reduced to a single set of chromosomes. So you can see that in each of these 4 cells now there are 2 chromosomes. Remember that the original parental cell had 4, that was the diploid number, and this has now been reduced to 2, which is the haploid number. So this was the double set of chromosomes, and now we have a single set of chromosomes, and of course a single set of genes. And you can see here in this final image we have the so-called tetrad of 4 haploid cells, which result from meiosis I followed by meiosis II. We're now going to go on and look at the consequences for these cellular actions at the level of genes and their alleles. There's a link to the next video. There's a chance to subscribe to the site. That's always greatly welcomed. And here's the full genetics playlist.